Okay, ladies and gentlemen, good day all. So in today's video, we are going to look at the concept of cloning. Please don't forget to subscribe, like, and turn on your notification so that whenever we upload videos, you can get notification. Introduction to cloning. Cloning is a process of creating a genetically identical copy of an organism or a cell. So when we said a cloning is a process that involves the creating an organism that is identical with the original organism or with the original cell. And this cloning, it has applications in medicine, in agriculture, and research. So one thing that I wanted you to understand, ladies and gentlemen, this cloning is very important in medicine, like for example, in personalized medicine, or in drug testing, as well as the vaccine development. You can get organs, you can get cells, specialized organs or specialized cells from stem cells, which can be used for many purposes. And cloning is also very important in agriculture, especially in creating genetically modified crops or organisms. And it is also very important in research, like in drug development, in vaccine development. Cloning is very important in that angle. Cloning raises ethical questions, that is, religious, social, and legal issues, or social, ethical, and religious concern about the diversity and the identity of the organism. So let's look at the example here. If you can see clearly from here, ladies and gentlemen, this is a DNA. So this is the original DNA, and this is the DNA clone. And if you can see, the original DNA and the DNA clone are identical. They are identical copies. So that is what you call cloning. So this is a typical examples of molecular cloning. And like, for example, in the case of reproductive cloning, this is a sheep, and this is a sheep clone. So you can create identical organism. You see the original sheep and the clone sheep, they look identical. So that is what you call clone. That is to create an exact copy or identical copy of an organism or a cell. Types of cloning. We have reproductive cloning. Reproductive cloning, it aims to produce a new organism genetically identical to the original. So in the case of reproductive cloning, you are producing an organism that is genetically identical with the original organisms, like the producing of a sheep from original organisms, like as shown in the previous slides. You have a sheep and you produce a sheep clone, and if you look at them, they are identical. So that is reproductive cloning. So then the next thing is therapeutic cloning. A therapeutic cloning, it focuses on generating tissues or organs for medical treatment. You can use embryonic stem cell, instruct them instead for, let's say, someone is suffering from kidney damage or liver damage, which of course will need an organ transplantation. So you can apply this therapeutic cloning to generate an organs or a tissues in the lab so that you can use it to transplant for the transplantation instead of harvesting organ from a particular individuals or from a particular or from a dead human. So you can just use stem cell technology by applying therapeutic cloning to generate organs or a cell or tissues to transplant it to the individual. Then molecular cloning. So in the case of molecular cloning, it involves copying a specific genes for research or medical purposes. Let's look at the example here. This is reproductive cloning. And as you can see from here, we have a somatic body cell and we have egg cell. So an egg cell from a lady can be isolated and the nucleus will be removed, and also the nucleus of the somatic cell can also be removed. And then the nucleus from the somatic cells will be placed into the egg cell. And then from there, you will have like a recombinant cell, which contain different nucleus and different content of the cell. And we call that clone. So this clone cell, you see they multiplied themselves and they give multiple copies. So it means that they are identical. So with this, the same clone cells, you can generate the entire organism. And that is what you call reproductive cloning. So at the end, you are producing an entire organism. So this is examples of animal cloning. And the importance of this animal cloning is very important to save endangered species. That is organisms that are almost or close to elimination on Earth. Because there are organisms that if you now check on Earth, their species are very scanty, meaning that they are endangered and there is a likelihood they may become extinct. So you can apply reproductive cloning to generate and regenerate the same of that organisms so that at least you can save them from becoming extinction. And you can use this reproductive cloning to copy elite animals. And then the next one is reproductive cloning. Is the starting point are the same, where you have the somatic cells, get the nucleus, fuse it with the nucleated egg cell, you have a clone cell, and from there, that clone cell can be used to generate a tissue or organs. So you can generate the entire cell or the entire tissue or the entire organ using stem cells or embryonic stem cells. And as you can see clearly, the importance of this therapeutic cloning is to make transplantable cells. So you can get a cells, you can regenerate a specific cells, which can be used for transplantation, and you can generate tissues or organs which can replace a damaged tissues, as you can see. And then the next one is transgenic techniques, where you use a specific gene of interest and you place it into a particular cell, and that specific gene will amplify itself. And at the end, this is how you generate genetically modified organisms like crops. So these transgenic or molecular cloning techniques is very important in research models or to develop a research model organisms or develop organisms that can be used in xenotransplantation. So these are the different types of cloning. So then the techniques in cloning, so we have somatic cell nuclear transfer, which is a common method used in reproductive cloning. And we have embryonic stem cells. Embryonic stem cells are the cells that are often used in therapeutic cloning to generate specialized cells or organs which can be used in transplantation or in research. And we have CRISPR. Advances in gene editing tools like CRISPR have enhanced cloning capacities because you can have a clone cell and you can use a CRISPR to edit the gene so that it will give you desired exactly the kind of organisms or the kind of genetically modified organism or the specific types of tissues or cells that you want to come up with. So as you can see, this is what you call somatic body cells. And in the somatic cell nuclear transfer, you can see this is a somatic cell body and we have an egg cell. So the nucleus from the egg cells is removed, and then the nucleus of the somatic cells is also removed. So you see the nucleus of the egg cells is removed, and then the nucleus of the somatic cells is also removed. And then the nucleus fused with the denucleated egg cells, and at the end we have a clone. So this clone can be used to generate the entire organism. So this is actually in reproductive cloning. And then in the therapeutic cloning, so you have the clone cells. So that clone cells can be instructed to differentiate into a specific cells or specific tissues or organs, which can be used for transplantation, especially in personalized and precision medicine. Ethical and future consideration. Cloning raises ethical issues about identity, consent, and the potential for misuse. The future of cloning includes possibilities. So actually, there is a lot of ethical and future considerations regarding the cloning issues because religiously, sometimes if there is this issue of cloning, sometimes you just generate an entire organism and there is a potential for misuse. If you are now a scientist and you realize that you can create the same organism, so why not you can decide to create Mukhtar Ali or you can decide to create any other organism. So this is some of the potential misuse that scientists and other people can actually make use of this cloning. So that is why there is a lot of religious, social, and legal concern. So the future of cloning include possibilities in regenerative medicine and conservation. Ongoing debate focus on the regulation and moral implications of cloning technology. So ladies and gentlemen, thank you for joining and don't forget to subscribe and join to my YouTube community. Thank you very much and see you next.